Everyone. My name is Oviya Ravi, and for this project, I was mentored by Lucy. I am currently presenting from Fremont, California. My project details the role of the protein P53 in mechanisms such as apoptosis, cell cycle arrest, and cellular senescence in cells that have been affected by DNA damage. The reason I chose this project was to learn more about P53 because of the extensive array of mechanisms that it plays a role in. Upon learning that P53 mutations are a major reason for which cancers arise, I was intent on learning more about why that was and how one protein is capable of being responsible for so many cancer diagnoses. First up, we have cell cycle arrest. For context, when cells divide, they undergo four distinct phases: G1, S, G2, and M phases. During these phases, the cell grows and produces various materials, replicates its DNA, continues to grow, and then divides its nucleus and cytoplasm, officially undergoing mitosis. Cell cycle arrest is when cells temporarily halt the cell cycle at G1, G2, or M phases, often in the setting of DNA damage. Cell cycle arrest typically occurs when there is repairable DNA damage, such as damage caused by ultraviolet light, ionizing radiation, oxidative stress, and more. Cell cycle arrest provides cells with the time to repair DNA damage and then proceed with the rest of the cell cycle. P53 plays a role in cell cycle arrest by increasing production of the P21 protein. This protein induces cell cycle arrest in the G1 phase by binding to cyclin E and cyclin D. This inhibits many target genes that are required for DNA replication, therefore halting the cell cycle. Apoptosis is a form of permanent cell death that is carried out if a cell is damaged beyond repair. This mechanism removes cells with DNA damage or other irregularities that could potentially become cancerous. P53's interaction with the BCL2 family of proteins, such as BAX and BAC, is one of the primary forms of apoptosis induction by P53. The ASPP family of proteins also plays a large role in activating apoptosis target genes of P53. Cellular senescence is an irreversible form of cell cycle arrest caused by P53. This mechanism puts cells in a state where they are not dead, but they no longer go through the stages of the cell cycle. They cannot grow or replicate. Although they are in a permanent state of arrest, they are still considered metabolically active and can secrete molecules in order to communicate with neighboring cells. Cellular senescence is linked to telomere damage and chromatin alteration, which activates the DNA damage response that activates P53 and arrests cell growth. Cell fate decision is how P53 chooses which pathway to follow upon responding to DNA damage. The process by which this is done is widely unknown, but there are speculations. One of these hypotheses is that the level of DNA damage a cell has is the deciding factor on which pathway to carry out. Others believe it is the type of cell responding to P53 activation or the site at which P53 is being phosphorylated. Now, why is all of this relevant? Close to 30% of all cancers have a mutation in P53. These cases have a 34% less survival rate compared to wild-type P53 cancers. Because of the immense role P53 plays in cell growth, cell division, and repairing DNA damage, P53 mutated cells have the ability to grow and proliferate uncontrollably, often resulting in tumorigenesis. Because chemotherapy often activates P53 to induce apoptosis, P53 mutated cancers are highly treatment resistant. Going forward, I believe there needs to be more research done on cell fate decisions because it would be crucial to understanding how to exploit the various mechanisms of P53. Understanding the differences in inducing cell cycle arrest and apoptosis could be extremely helpful in the development of therapeutics. It is also important to study the different ways to induce apoptosis and cell cycle arrest in the absence of P53 in order to better the prognosis of patients with P53 mutated cancer. Thank you for listening.